So we have a couple of questions so far. So the first one is for Dr. Fernari. There was a question about the character change at advanced stage of glioblastoma. What can be done to cope with the aggressive changes in the patient's characterization?
I think that's probably a question that Ingo or Al could address. So Lisa was asking about aggressive asocial changes in patient's character associated with the tumor.
Okay, thank you.
Well, character change in terms of as expression of symptom from the tumor may be related to the location of the tumor, related to whether the tumor has moved from one location to another location. That will express a new symptom, a new character, and that although we know that in GBM most of the time the growth of tumors is in the locale the original tumor is, but the tumor, as the tumor can progress, it may become more aggressive and more invasive to move to another location, causing a new set of symptoms. And that change of character is related to the biology of the tumor. And that's also why we want to study the biology of the tumor in each step of the way, so that by knowing the mechanism that behind this change of behavior, we'll be able to apply the right treatment to this new behavior.
Okay, thank you so much. So I'm just scrolling down. The next question is, what are some of the inhibitors being developed for combating IDH1? And secondly, have we seen resistance with the early testing using MRS, resistance against the inhibitors, that is?
Okay, I can speak to that a little bit. So the IDH mutant tumors, gliomas, they have certain unique features. They show greater degree of methylation. We call it the hypermethylator phenotype. And they also show differences in differentiation markers. So there are several approaches that are being pursued right now to target IDH mutant gliomas. The most direct one is the development of inhibitors that bind to the mutant enzyme itself. That's the most intuitive, perhaps, choice. And there are several companies that are developing these right now. And actually, just as on a side note, IDH mutation happens not just in glioma, but also in other human cancers. For example, in AML, acute myeloid leukemia, also in certain sarcomas, soft tissue sarcomas, cholangiocarcinoma. And I'm mentioning this to you because the inhibitor of mutant IDH, or the development of these drugs, is most advanced right now for IDH2 mutations in leukemia. And that was reported at the AACR meeting earlier this year, where these mutant IDH inhibitors have shown very promising results in phase 1 in acute leukemia. So this kind of drug, these are mutant selective enzyme inhibitors. There are other ways of thinking about this problem, and that would be viewing the mutant IDH as a sort of differentiation block, cellular differentiation block. So the idea here would be to restore the ability of tumor cells to differentiate into normal cells. So there are thoughts of trying to approach this through changes in the epigenetics of the tumors. So these are drugs that do not directly bind the mutant enzyme, but they interfere or block enzymes related to DNA methylation. So those are, I think, the two main approaches right now. And the mutant IDH selective inhibitors have begun to be given now to patients with glioma. There's a phase 1 study that's currently open that I'm aware of. We're one of the sites, and it's too early to comment on whether there are changes. It really is brand new, but I think at the next conference call, maybe we can update you on that. But they have begun to be in the clinic. That's the exciting part.
Great, thank you so much. So the next question that just came up, given that IDH1 in brain tumors responds differently to drugs and is different than other cancers, such as lung, is the assumption that new drugs would be needed or that other existing drugs would be effective?
Well, so as of today, there are IDH2 inhibitors only in clinic already for which there's data available. And those are the mutations in leukemia primarily. So yes, you're right that for the glioma, almost all the gliomas have mutations in IDH1, not 2. So there will be different drugs than what's being used in leukemia, and they will be developed likely in a way that pharmacologically differs in order to improve their penetration in the central nervous system. That's a big component of this effort.
Great, thank you. So I have a question that came in privately, asking us to talk more about the unique qualities of having these various cores working together, collaborating together. How are we thinking that that's going to accelerate the process of therapeutic development? Dr. Young, do you want to head that one?
Well, the idea that we had when we were discussing what can we do that we can really make impact in in GBM, which if you look at it compared to other difficult cancer, such as melanoma, such as non-small cell lung cancer, some breakthrough has come to those cancers. But in GBM, we're still looking for some more effective treatment that's better drug. And when we put our head together, we say, well, maybe one way to do that is that we can have a group of investigators. Now, these experienced investigators, if we put them together and tell them, say, this is a problem that you need to solve, and you can solve it together, and we can identify which way to do it, which direction we need to move, where are the most advanced lead that can help us to change the curve, and we go in that direction together. And this is how the 4 core idea come about. We need to understand the biology better, and we need to have better drug, and we need to have a way of identify the patient so that we can identify patients that will be sensitive to certain drug, will develop side effect from certain drug. We want to know ahead of time so that we do not cause harm and we can achieve maximum effectiveness. But what the most important thing that we think is that by putting a group of people together that work well together, that can share idea together, can openly discuss information that generated from each laboratory together, then we can synergize the effort there. Instead of each person work in a different environment, kind of in a vacuum or in isolation, we put a group of highly effective people together, talk to each other all the time. We think that will synergize the effect. And that's what this consortium concept comes up.
So much. There's an additional question. What is or will be the connection between Defeat GBM and the pharma companies? And Dr. Young, I think you're also best equipped to handle that question. It's an interesting relationship that we have with the pharmaceutical companies right now.
Well, we are developing relationship with the pharmaceutical company because when we talk about and when we understand that we do not have a good drug for brain tumor, not that there are no drugs out there, but on the other hand, we need to understand that if we can get the involvement of the company ahead of time, while the company are developing drug for other cancer, that same drug can be applied to brain tumor. And if we as the Defeat GBM as a consortium can work with a company in the very early stage of the development of a drug, we can actually take over some of the risks that the company may have to encounter to develop a drug or to look at a drug for a small tumor like GBM. And if we collaborate with them as a group, Defeat GBM, and we take the responsibility of looking at it with the right model for GBM, it will allow them to give us the drug to look at. While they are looking at the drug for lung cancer, breast cancer, we were looking at it for GBM. That's the kind of relationship we want to develop with the drug company. And so that the company can give us the early drug that we look at, then we have a chance to investigate a larger number of drugs as you going to the pipeline of drug development. And we now is talking to several companies. Many of these pharmaceutical companies have a large portfolio of drugs, and we set in relationships so that we have a chance of looking at that portfolio earlier while they are looking at a portfolio for other cancers.
So, and speaking on behalf of the National Brain Tumor Society, I think one of the things that have been most exciting for us with Defeat GBM has been our changing role as a nonprofit, a patient advocacy group, and our relationship with not only pharmaceutical companies, biotech companies, and the research institutes themselves. And so you'll often hear that we're now partners with these various stakeholders, these various groups. And that is certainly the case with the pharmaceutical companies, as Dr. Young shared. And we have created or are creating what we're calling a black box so that our researchers can have access to these compounds at the moment that they need them. So it is quite an interesting partnership and one that we're very proud of. I also want to expand that because one of the questions that came in was regarding one of the slides that Dr. Young had about the Cure GBM LLC and the fact that this was a $2,500 initiative. The National Brain Tumor Society has actually affected a new entity to warehouse Defeat GBM, and we've created an LLC, again, towards that systems change, that need to create a different dialogue with the research community. While we are investing a minimum of $10 million towards this initiative, we're leveraging our relationships with the various institutions, and they are also contributing a dollar-to-dollar amount. So that shows certainly their willingness and their investment and acceptance of this program as and their leadership in trying to improve the lives of our GBM patients, and we're grateful for that. Do we have any other questions coming up? I don't see any. We're nearing the end of our webinar time. I want to thank everybody for their participation. Dr. Young, Dr. Mellinghoff, Dr. Finari, thank you so much. Chisa, I see one more question. What is, oh, we do have one more, forgive me. Is the Defeat GBM initiative working with the New York Genome Center's efforts that is focused on GBM?
Yes.
Ingo, do you know that?
Yeah, of course. I mean, we have Bob Darnell here, who's a brilliant researcher and friend, so we'd be foolish not to engage him. You know, this is great. I think this is one of the greatest strengths that several of us, we tap into different resources and we can engage as needed, and Bob Darnell at the Genome Center is absolutely one of them. So we're very blessed to have that resource, yes.
Yes, and one of the powerful strengths of Defeat GBM is that we have built a structure that allows other folks to come in, other researchers and institutions, and we're happy to be working with them. I want to thank the presenters again for your time. I want to thank the audience for your support. We take our commitment to you very seriously. Thank you very much. Have a wonderful day.